unreasonable. And in fact, if anything, coffee protects from pancreatic cancer quite paradoxically. Then obviously this reduces the credibility of epidemiology as a discipline, and there are many people who say, well, you epidemiologists, one day you say black, the next day you say red, the third day you say green, so no, we can't really believe you anymore. Obviously there is a waste of public resources in terms of putting money to replicate results that are wrong. And well, as I said, those that are generated just by chance will eventually be corrected by replication. I will show one example of this. However, this entails inefficient use of resources. This is, well, it's a complicated slide, but it doesn't matter. I mean, the point is that we try to do an empirical evaluation of false positive results in the domain of occupational epidemiology. And this is basically what we did. Well, I can probably go. No, this was some previous example in genetic epidemiology. What we did was to work on occupational epidemiology because we could use this WHO, IAC is the agency of WHO for cancer research, evaluations that are sort of benchmarks, which sort of make the point of how strong the evidence is for this or that chemical at some point. So we look at the chemicals that were classified as intermediate, suspected carcinogen, and see what happens over time as evidence accumulates, to see whether the early results that make the chemical to be classified as suspected carcinogen were confirmed or not, whether first results were positive association, were false positives or not. And we did it for 23 associations for one particular occupational carcinogen and one particular cancer, to 150 different combinations, and in fact, 150 different studies dealing with these 23 associations. And in fact, for 12 of them, which is a bit half, or roughly half, the evidence decreased over time. For 6, there was no change, it was weak and remained weak, and only for 5 of them, the evidence grew, became stronger, and were recognized as carcinogen. This is just one example of a false positive. It's acrylonitrile with lung cancer. There were 16 studies with different populations of workers exposed to acrylonitrile for a total of 300 cases of lung cancer. This is the results of the first study, published in 1978. You see this is on a logarithmic scale. So the relative risk was about 4. So people involved in acrylonitrile, exposed to the acrylonitrile in the workplace had 4-fold increased risk of lung cancer. But then in the subsequent years, there were more and more studies published, up to these 16 studies. And each time, the new estimate here, the new point, is the average of what was done up to then. So in whatever 1982 or whatever it is, there was a second study which reduced the average from 4 to 2. So obviously it was a result of 0 because the first was 4 and then the average was 2. And then if you go down, you see that the evidence became weaker and weaker. So there were more and more null study or negative studies. And at the end, when the last evaluation was made, which is now already 10 years ago, but we didn't follow because at that time there was no formal evaluation. There was basically very little left. There was a very, very small increase. So this is an example of these early results probably being, well, not probably, being clearly not representing truth, but just maybe they were higher there by chance, or maybe there was something particularly strange in that factory, I don't know. The point is that, the big story of acrylonitrile as a cause of lung cancer, which was generated by these early results, were not confirmed. And another example, welding, fumes, and lung cancer. Here there are 11 studies, up to 88 only because there was no further evaluation after that. And in that case, you see that, there was a small increase, but it was confirmed. The more studies, the smaller the confidence interval because it's more and more precise, but it stays there. So maybe it's something true. So the two analyses convey a very different message in a way, and we did it for many others. Okay, these are some of the limitations of our exercise, but I don't think we need to discuss it. This is another example. This is related to something closer to what you are involved. It's a study which was published in '93, linking breast cancer in women to a serum level, a blood level of DDE, which is the main metabolite of DDT. And this was done in New York, These were a group of women who donated blood and then were followed prospectively, as we discussed before. And after some time, some of them developed breast cancer, some didn't. Investigators went back, got the blood sample stored in the freezer, analyzed the level for those who developed cancer and those who didn't develop cancer. And the level of DDE was split into 5 categories. These were the lowest category, this was the highest category. There was this dose response. Not completely monotonic, but suggesting that the higher the level in blood of DDE, so the higher exposure to DDT, whatever this means, the higher the risk of breast cancer. So big story, DDT is one of the major causes of breast cancer, etc. This was published in '93, and this is the same result at the top category in the previous slide. Again, there was a call for new studies replicating. We found the major environmental cause of breast cancer. There were another 1, 2, 3, 4, 5 studies published in the following decade, basically. And you see that, again, the cumulative evidence went down to relative risk of 1, so no excess. So again, the results of the first study in New York was a false positive. And this has been shown. Now nobody talks about DDT as a cause of breast cancer anymore, I guess. So all this is sort of examples. The framework for this work is, as I said before, these evaluations that were done by WHO on the evidence coming from epidemiology and other systems, not only human data from epidemiology, but also sort of integrated animal and mechanistic data. And if epidemiology is very strong, like in the case of tobacco smoking and lung cancer, etc., the agent is classified in what is called group 1, established carcinogen. There is another possibility to classify in group 1 if the agent is an animal carcinogen from animal experiments, and also there are strong mechanistic data suggesting that the mechanism may operate also in humans, like generating DNA adducts or generating some alterations in some gene or whatever. And there is some evidence from epidemiology. That's the reason of the dotted line. I mean, maybe not very strong, maybe only one study. But if all this goes together, then the compound can be classified in group 1. Yeah, I do quickly through. When there is no such mechanistic support of data, the classification is called group 2A, which is probable carcinogen. It's called probable carcinogen. And when there is only animal data, with no human data, no mechanistic data in humans, the classification is typically what is called group 2B, possible carcinogen. Now, what's the situation for pesticides? There are 3 pesticides in group, 2 pesticides and 1 contaminant, dioxin, in group 1. There is the arsenic, as used in insecticide, ethylene oxide, and as I said, DDT. So basically, none of this is used anymore. DDT, TCDD should not be present or should be at extremely low level or whatever, and the other 2 are no longer used. There are 3 in 2A. This category, if you remember, where there is, just to remember, 2A means that there is evidence from experimental setting, from animal data, and some human data. And there are captafol and EDB, ethylene dibromide, are in group 2A, and also the broad category of application of insecticides without, specific, being able to identify specific agents. And these are the year when these evaluations were done. That's the typical example. Ethylene dibromide has sufficient evidence in animals and some evidence inadequate but not very strong in humans. And then there are 19 pesticides in group 2B, which, if you remember, is basically those who have sufficient evidence in animals and basically no data in humans. And you have our friend chlordecone is here, was classified in '87, chlordane, chlorophenoxyherbicide, DDT, DCBP, heptachlor, and a few others. And they are all been shown to be carcinogenic in animals, but there are no supporting human evidence. I think I can skip. These are the core studies and the studies on pesticide, etc. I think this we can close. But just to I have no time to go into the detail of the review of the epidemiological studies on pesticides beyond, the specific examples that I described. But in general, the problem with the interpretation of studies, and now there are two large studies in the field. One is the one mentioned by Mr. Bousquet, which is conducted in France, in Cannes, in Normandy, and in Bordeaux, well, in fact, in several areas under these two columns. And there is a study of the American agricultural study, so-called agricultural health study in the US done by NCI. But in general, what is available in the literature, most of the studies are too small to detect weak effects, so they are really not able to tell us, they are able to exclude a very strong association, but they are not able to tell us, whether there is an association at the lower level of relative risk. Most of them tend to have multiple comparisons, many different pesticides, many different outcomes, some positive association just by chance. And there are issues in terms of bias, in particular recall bias and publication bias. Publication bias is a very interesting issue. Again, I have no time to discuss it, but we can maybe address it during the discussion. So, in conclusion, when we think about pesticide and cancer, we really feel into this domain of weak association, which is a major challenge to epidemiology research. I mean, it's easy to identify some factor that causes an enormous number of disease in the population, an enormous increased risk. Fortunately, we are not there. That's very fortunate, in fact. So we are confronted with a much more challenging situation. False positive results is a major issue when people look for weak associations, because they have to elaborate many, many different, disease exposure association to identify. Therefore, in my view, adherence to high epidemiological standards in the design of the studies, analysis, reporting, and interpretation is a possible way. In terms of the more specific issue of pesticide and cancer, there are a few agents that are known or suspected, highly suspected carcinogens. They are basically being regulated. There are several others that are suspected and need to be studied, and fortunately people are studying it, and it's possible, I think, to study it in the right way. In general, however, the current evidence does not suggest, at least in my interpretation, that pesticides represent a major cause of human cancer. This doesn't mean that there are no risks whatsoever, but it's unlikely that this is indeed the case. And I stop here. I hope I didn't go too much in time.
Thank you.
Okay.